Now for something really special, we begin Chapter 8 of our discussion of exercise physiology. How important is skeletal muscle to the discussion or study of exercise? It's just a lot of fun too. It reveals a lot of properties of our bodies in terms of exercise and why things are the way they are. So let's take a look at Chapter 8, Part 1. So general functions of skeletal muscle. You've got to be able to move the skeleton. That's what exercise is, right? So that's pretty basic. When we're not exercising, skeletal muscles are the basis for postural support. In other words, being able to hold up our skeleton in the positions that we want to retain for various functions, interacting and eating and whatever. Heat production. We are temperature-regulated organisms, and the source of heat is every single cell in your body that's hydrolyzing ATP. But your muscles produce a lot of that heat during various activities, and in fact can be called upon for shivering to produce extra heat in case the body temperature is starting to fall. Well, we have some terminology to describe various muscle contractions that will help us be efficient in our discussions. Flexor muscles are ones that either decrease the angle between bones at a joint, such as if you're flexing your forearm, the angle between your forearm and your upper arm is diminishing. Or we also use that term to describe the anterior movement or forward movement of your arms and legs at the shoulder and hip. These words we have to just accept. Extensors are muscles that increase the angle between two bones at a joint, or that move the arms and legs posteriorly relative to the resting position of the body. It was called anatomical position, standing straight up with your arms at your sides. Abductor muscles move the arms and legs away from the midline of the body, and adductors return them to the anatomical position, to the straight up and down position, or at your sides in the case of the arms. Here's an illustration real quick. So we have flexion of the arm, extension of the lower leg, flexion of the lower leg. So we see this angle decreasing, this angle then increasing between the lower leg and the femur, the thigh bone. Flexing the arm, moving anteriorly, extending the arm at the shoulder, moving posteriorly. Hyperextension. Once you pass this resting so-called anatomical position, it's hyperextension. Abduction, again, moving the limb away from the body's center line. Adduction, returning it. Circumduction is just a rotational movement. There are certain exercise applications that involve circumduction. Okay, let's talk about the anatomy of muscles and muscle cells. Muscle cells are packaged together to form organs called muscles or skeletal muscles, and the entire muscle is surrounded by a connective tissue covering, a jacket. That's the epimysium. And it's continuous with the tendon that attaches it right to the bone. Within the muscle are bundles of muscle cells called fascicles, and each fascicle or bundle is wrapped by another layer of connective tissue called the perimysium, right? Collagen fibers. And then each individual muscle cell within the fascicle has a fine connective tissue covering called the endomysium. So those are some general coverings of muscle cells, and all of those connective tissue layers are continuous with the tendon, very firm anchoring to the bone. The basement membrane is a sort of like a mat, a dense, thin, dense mat of proteins produced by the muscle cell that's just beneath the endomysium. So the outer, that fine connective tissue wrapping of the cell, and then under that is what's called the basement membrane, and then finally the cell membrane, the actual phospholipid boundary. It's called the sarcolemma. When you're talking about muscles, there's a couple of important prefixes that designate muscle. Sarco means of muscle, and myo means of muscle. So sarcolemma is just a fancy word for plasmalemma, the plasma membrane. So here's a cartoon to illustrate. We have a muscle organ wrapped up by the epimysium here, the connective tissue connected to the tendon. And then we have bundles of muscle cells that comprise that muscle called fascicles. Each fascicle is made up of a number of muscle cells. The fascicle is wrapped up by the perimysium, this connective tissue covering. that each individual cell is wrapped up by the endomysium. We see pulled out an individual muscle cell out of this fascicle, so we can take a look at it. We see the endomysium, but we also see some organelles within that muscle cell called myofibrils. So the cytoskeleton of a muscle cell is 90% or 90-something% of its entire composition, and the myofibrils are the units of cytoskeletal arrangement. Satellite cells are not muscle cells, but they lie underneath the endomysium. There's some cells almost like stem cells. They're like some kind of primordial cell that's there on the surface of the muscle cell, and they're thought to be important in repairing damaged muscle tissue. They can morph into they can either insert themselves essentially into a muscle cell to add more nuclei, as we'll see, or they can actually, it's thought, produce new muscle cells to replace cells lost by injury. So, what they mean by the myonuclear domain. Skeletal muscle cells are long cylindrical cells that span the entire length of a muscle. That's crazy, right? Cells are microscopic, and yet skeletal muscle cells span the entire length of the muscle, whether it's your thigh or your arm. So that's pretty impressive. And in order to be able to do that successfully, there has to be a bunch of nuclei, because nuclei are the origin of information for making proteins, right? The chromosomes. And each nucleus can only kind of supply that information by way of messenger RNA for a certain volume of the cell. And then you need another nucleus and another nucleus for these incredibly long cells to have enough information. So that's called a myonuclear domain. The one nucleus and the amount of cytoplasm it can really inform. So there's a lot of nuclei there, and it's thought that satellite cells can contribute nuclei to the muscle cell as needed, even during adaptation to exercise. Okay, so back to our myofibrils. Those are the organelles that contain all the cytoskeletal elements, and the main ones are actin, the thin filaments, and myosin, the thick filaments. And they just interdigitate to produce the arrangement of, in the myofibril. A sarcomere is the simplest unit of such arrangement, the simplest unit of a muscle cell, the simplest, smallest functional unit, because sarcomeres have all the hardware they need to actually contract and shorten. So let's take a look at some sarcomeres. Here's a myofibril pulled out from a muscle cell, or muscle fiber it's sometimes called. It's an unfortunate use of terms in my opinion, but a muscle cell is also called a muscle fiber. And the myofibril is constructed of repeating units of the arrangement of these cytoskeletal proteins. The blue you see there is actin thin filaments, and they surround or interdigitate with what's drawn here in red, myosin thick filaments. And every one of these repeating units is called a sarcomere. Right? There's a boundary to which the actins are attached at each end of the sarcomere, the Z line. And then there's these myosin filaments in there. It so happens that if you look at a skeletal muscle cell in the microscope, areas where the myosins and actins overlap produces the densest arrangement of proteins. You can see all the filaments here in this end-on section, but wherever the proteins are overlapping, that's the most dense area, and it makes a dark line in the microscope. And wherever there is not overlap, here we're looking at this area here with the Z line and just actin filaments, it makes a light-colored band. So when we look at skeletal muscle in the microscope, it's striated, it's striped. And that's because of these differences in density of the cytoskeletal proteins at particular muscle lengths. A couple of other organelles or features we need to know about a muscle cell to begin to understand how it works. The Sarcoplasmic Reticulum is just the fancy name, again, the sarco, muscle related name for the endoplasmic reticulum. All cells have it. Muscle cells are no different. In the case of muscle cells, not only is it important in protein synthesis, like in all cells, it's the site of calcium storage, the reservoir of calcium. There are no calcium ions in the cytoplasm or the sarcoplasm at rest, only during contraction. So that's the source of the switch for activating contraction. The sarcoplasmic reticulum has kind of bulbous units, as we'll see, in kind of a repeating arrangement. Those enlarged units where there's a lot of volume of calcium solution is called the terminal cistern. Cistern is just an old-fashioned name for a tank of water in your basement where you collected rainwater before there was plumbing and you had to collect your own water to use for cooking and washing. A cistern. That's a pretty fun historical reference. That word crept into the nomenclature of muscle organelles. And transverse tubules, the sarcolemma or the plasma membrane forms little holes and passageways right down through the muscle cell. So the plasma membrane passes right down through the deepest core of the muscle cell on a repeating basis. Let me show you what I mean. Here's a cartoon of a skeletal muscle fiber or cell, and they've drawn the sarcolemma in pink so you can kind of see that these so-called transverse tubules are actually made up of the cell membrane, phospholipid bilayer that passes all the way through the cell. And snuggled up against those transverse tubules are the terminal cisterns, or terminal cisterna, in the old-fashioned Latin-y kind of form, of the sarcoplasmic reticulum, which is drawn in green. So those are essentially membranous sacs full of calcium. And here's the nucleus of the muscle cell. Here's the myofibril, made up of all those actin and myosin. You can see, if you look carefully, it's got some sarcomeres drawn in there. So that's just the basic construction of a muscle cell, and we'll come back to the importance of all of these features when we talk about how the thing gets activated and interacts. How do we activate a skeletal muscle cell? Well, a motor neuron carries impulses from the brain, from your desire to contract the muscle. Impulses are nothing but action potentials which sweep down the axon. Wave of depolarization reaches the axon terminal, and the axon terminal where it's attached to a skeletal muscle cell is called the neuromuscular junction. The place on the muscle cell where the axon terminals are attached is called the motor end plate. It's the sarcolemma where there's a neuron to talk to it and stimulate it. When you think of motor neurons, each motor neuron, nerve cell that comes down from the brain to the spinal cord and out after a synapse out to the skeletal muscle, each neuron innervates more than one muscle cell, or activates more than one muscle cell, and it's permanently affixed to a specific subset of muscle cells throughout the muscle, and that motor neuron, each motor neuron, and the muscle cells that it innervates or attaches to is called a motor unit. We'll see that is how you activate muscle cells in groups called motor units. The place where the axon contacts the muscle cell, or nearly so, there's a bulbous enlargement of the axon of the neuron, the axon terminal, and that sits very, very close to the muscle cell membrane, but there's a little gap in there called the cleft. The neuromuscular cleft, or the synaptic cleft, a space that's between them with some fluid in there. When the motor neuron is activated, it releases a neurotransmitter. The neurotransmitter in the case of neuromuscular junctions is acetylcholine. Acetylcholine is released by exocytosis. It diffuses across the cleft and binds to receptors on the motor end plate, on the muscle cell. Now, the muscle cells start out with a negative electric charge inside the cell relative to outside, so it's negatively charged inside, positive on the outside. And when the acetylcholine binds to its receptors, the receptors are channels. They pop open and let sodium ions rush in. Right? Sodium is highly concentrated outside the cell. And if you open a sodium channel, sodium ions, which are positive, rush into the cell from high to low concentration, and if you're letting positive charges into a negatively charged area, they're going to dissipate that negative charge, and that's what happens. It's called depolarization. That's the stimulation of a muscle cell, depolarizing the neuromuscular junction by the action of acetylcholine. That's called an end plate potential. What's the change in electric charge on the motor end plate where the motor neuron terminates? If there's enough depolarization at the neuromuscular junction, it's going to trigger an